Hello, everybody. Welcome back. This is the first of our mini lesson from chapter 7, which is going to be kind of our microbial genetics section. In this little mini lesson, it's just going to be a little bit of background information and then focusing on some specific details of prokaryotic genomes. So we'll do a quick review of DNA and RNA and then see how things are slightly different in prokaryotic cells. Okay, so here is a diagram of DNA and RNA. So just to remind you, DNA stands for deoxyribonucleic acid. It is a double helix made up of sugar phosphate backbones, which is represented by the blue ribbony parts on this diagram. We'll see the actual sugar phosphate backbone on the next slide. And then the middle, the rungs of this twisted ladder form, if you wanted to call it that, are made up of 4 nitrogenous bases: A's, T's, C's, and G's. So you can see those listed there on the left. And then compare that to RNA, which stands for ribonucleic acid. This is a single-stranded molecule with a sugar phosphate backbone. The sugar is ribose, sugar instead of deoxyribose, like in DNA. And it also has 4 bases kind of coming across. It doesn't complement to anything, like the DNA has the complementary sides. And the bases are A's, U's, C's, and G's. So it has a uracil in place of a thymine base. Now, in eukaryotic organisms, the DNA was housed in the nucleus, and the RNA, the mRNA, was transcribed in the nucleus, traveled out to the ribosome, which was made up of ribosomal RNA, and the tRNA was used to bring the amino acids to the ribosome. So that was how you learned it in general bio with your eukaryotic cells. Prokaryotic cells have the same molecules, the DNA and the 3 different types of RNA. There's just not any division location-wise because there's no nucleus. So DNA and RNAs, all the RNAs are kind of hanging out in the cell in that space. So taking a closer look at the nucleotides and the sugar phosphate backbone, we can see the red circled structure is one nucleotide, which is made up of the base, either the A, T, C, or G in DNA, the sugar, this is a deoxyribose sugar, and then the phosphate group. So when in the last slide, that blue ribbony part was talking about the sugar phosphate backbone, it is phosphate, sugar, phosphate, sugar, phosphate, sugar. And so it kind of does this zigzag between the sugar and the phosphate. So that's why we call it the sugar phosphate backbone. And then sticking off of each sugar towards the center of your double helix, right? So if this is your sugar phosphate backbone, and this is the other sugar phosphate backbone, your bases are sticking off complementary to each other thusly, okay? So thymine, adenine, cytosine, and guanine. If we were to have the complementary strand of this short little piece of DNA, we would have an adenine, a thymine, a guanine, and a cytosine, right? Double hydrogen bonded there between the corresponding base pairs. All right, so that is our nucleotide structure, a little bit zooming in to the specifics. Also, just to point out before we move on, the 3' end and the 5' end. So those came into play when you were learning about DNA replication. Again, we're not going to be covering those topics in this chapter. They are listed here, the DNA replication, transcription, and translation. So if it's been a while or you're a little bit rusty with those topics, please make sure you go back and read through those. However, I'll be focusing just on the selected pages that's listed in the syllabus and what we cover here in the lecture. But reviewing those things, if you're rusty on the DNA replication, transcription, translation, might be helpful for you when we move on to the other parts of this chapter and other chapters as well. Okay, so that's just our basics, DNA and RNA. And now if we take a look at some specifics for the prokaryotic chromosome, we can take a look at those. Bacteria and archaea, our prokaryotes, have a single circular chromosome, whereas humans have 46 individual linear pieces, just one long string of DNA. Bacteria have a circular chromosome. So in this big green kind of bacteria picture here, our one circular chromosome is shown here. Now, it's not to scale, obviously, because there'd be a lot more DNA there. And this particular picture over here This is the bacterium that's been broken open, and all the squiggly lines right here, all of these, that is the chromosome kind of spilled out of the cell. So as you can see, it's not just one circle like they're kind of showing in this green bacterium over there. It's really going to be folded and crammed and smushed into the nucleoid region of the bacterium. It's relatively large compared to the size of the cell. So that's where all of the required genes are located for the bacteria to be the bacteria. But prokaryotes also contain accessory DNA, if you will, and these are in the forms of what we call plasmids. So plasmids are small circular pieces of DNA that do not contain required genes. They kind of contain bonus genes, extra genes that are not needed for the cell to be the cell. So some of the genes that are found on plasmids, we have the fertility factor. We'll be talking about that when we get to conjugation at the end of this chapter. It allows for a cell to produce a conjugation pilus. We saw that back in chapter 3 for horizontal gene transfer. Some of the plasmids contain resistance factors, which are genes that when expressed give the bacteria the ability to resist antibiotics. So it gives them, they have whatever the resistance factor is. Maybe they can neutralize the drug, or they can pump the drug out, or they can use a different pathway, or they can just get around the way that the antibiotic drug would work. So those are called resistance factors. They might also have bacteriocin factors, which are kind of like bacteria killing weapons. So they have the ability to compete with their neighbors. So bacteriocin means to kill bacteria factors. And then there might be some virulence factors. So something that the cell would have that when expressed would make it more prone to causing disease. So that might be a capsule, it might be a toxin, maybe some kind of slime layer or fimbriae or conjugation pilus to allow it to transfer those resistance genes or something like that. So the chromosome carries all of the genes needed for the cell to be a cell. The plasmid carries specialty genes that are not required. They're kind of the bonus genes, okay? Now, not all cells have to have a plasmid. They may or may not. It's optional. But when they do, and they express those genes, then they would be able to carry whatever traits are carried on those plasmids, okay? All right, and then lastly, just to remind you of what we call the central dogma of gene expression, kind of the role of DNA and RNA and proteins, is DNA codes for instructions for proteins. So all the DNA has, it's like the blueprint for the cell. It goes through the process of replication when it makes more of itself in preparation for binary fission in a bacteria or mitosis in a eukaryotic cell. In the process of getting ready to transcribe a particular gene, we go through the process of transcription and we produce mRNA. Well, all the other types of RNA are produced by transcription, but for gene expression, that mRNA has the codons, the code for the particular amino acids in the process of translation, that the end product is going to be a protein. Okay, so just you should be comfortable with this process. DNA codes for things, transcription kind of copies that code, and translation expresses that code into a product in the form of a protein. And replication is just the DNA is making copies of itself in preparation for division. Okay, so that's our little review of DNA stuff. Again, if you are a little bit rusty or uncomfortable with this idea of the central dogma, specifically replication, transcription, translation, read through these intermediate pages that I'm skipping over in the syllabus. You'll see them kind of outlined there in the text. Review that, send questions, come to office hour, because what we're going to be doing next is jumping over that material in the textbook and getting into how prokaryotes express their genes a little bit, especially in the process of operons. All right, so I will see you next time.